All living organisms are composed of one or more cells. Your own body is composed of tens of trillions of cells, such as these live skin cells. Let's zoom in on one of these animal cells for a closer look at the amazing world of cell structures. Extracellular matrix, or ECM. As we approach a single cell, we see it is surrounded by an extracellular matrix, or ECM. The ECM consists of carbohydrates and protein fibers, and functions primarily for protection and support. Plasma membrane. Below the ECM, we encounter the plasma membrane that separates the parts of the cell outside the nucleus, called cytoplasm, from the cell's outer environment. The plasma membrane closely regulates what enters and leaves the cell. The plasma membrane consists of a fluid phospholipid bilayer embedded with proteins. Transport proteins move substances across the membrane, as shown here. The fluid state of the membrane allows lipids and proteins to move laterally, and sometimes even flip-flop. Cytoskeleton. Look at the vast cytoskeleton, a complex network of protein filaments and tubules that extend throughout the cytoplasm of the cell. It supports cellular movement and structure, as well as transport within the cell. Two major components, microtubules and microfilaments, are shown here. In animals, microtubules sometimes form a spindle apparatus, which plays an important role in separating duplicate chromosomes when cells divide. The nucleus. Let's now move to the control center of the cell, the nucleus. The nucleus is separated from the cytoplasm by the nuclear envelope, a double membrane with numerous pores. Let's dive into the nucleus through a pore. Inside the nucleus is genetic material called DNA, which combines with stabilizing proteins to form chromosomes. When a cell is dividing, the chromosomes are highly coiled into short, thick, rod-like structures. DNA stores the instructions for making most cell proteins, as well as specialized RNA molecules. The instructions for making RNA and/or proteins are coded in the sequence of base pairs along the DNA double helix. Protein synthesis. How does DNA make proteins? It is a two-step process. In the first step, called transcription, an enzyme called RNA polymerase attaches to the DNA and transcribes it into a specialized RNA molecule called messenger RNA, or mRNA. The mRNA contains information for the amino acid sequence of a protein. mRNA is then transported out of the nucleus into the cytoplasm. The second stage of protein synthesis, called translation, begins when a structure called the ribosome attaches to mRNA. The ribosome moves along the mRNA, aiding in the assembly of amino acids to produce a protein. Protein trafficking. Some proteins are made in the cytoplasm by free ribosomes, as you just saw. Other proteins are made by ribosomes bound to a structure called the endoplasmic reticulum, or ER, an internal network of membranous sacs. The ER with ribosomes is called rough ER, while ER lacking ribosomes is called smooth ER, and is used for making lipids and carbohydrates. Those proteins that are made in the rough ER may be modified and then exported to their next destination, the Golgi complex. The proteins are packaged inside transport vesicles that pinch off from the ER. These vesicles travel along the cytoskeletal tracts. They carry the proteins to a stack of flattened membranous sacs called the Golgi complex. Here, the vesicles fuse to the Golgi. Here in the Golgi, enzymes inside may modify the proteins further. For example, a carbohydrate group may be altered, or the protein may be cut and then packaged inside vesicles, which carry them to their final destination, which may be within that cell or out of the cell. The mitochondria. In order to carry out activities, cells require a continual source of chemical energy in the form of ATP. ATP is made in the mitochondria by a process known as cellular respiration. The main ATP-producing machinery is found along the inner membrane of each mitochondrion. As we end our tour of the cell, keep in mind that we have briefly described only a few of the most important structures and processes that take place inside this amazing microscopic world.